Hello, grade 9. Today we're going to start chapter 8, Conformed Reproduction of Genetic Information, activity 1, Transmission of Genetic Information. The objective of this activity is to define mitosis, to describe the mechanisms that allow the body cells to possess the same number of chromosomes and the same genetic information. So, as you know, the egg cell or the ovule, having 23 chromosomes, is fertilized by a sperm cell with 23 chromosomes. And the result of this fertilization is a fertilized egg or a zygote with 46 chromosomes. Then, this zygote develops into an embryo and finally into an adult organism. How? By a process that we call mitosis. So the zygote divides many times, producing all the body cells. They all carry, except the gametes, the same number of chromosomes and the same genetic information. How is this possible? It's possible through the process of mitosis. What is the importance of mitosis? Every somatic cell in an organism body undergoes mitosis, and this includes skin cells, blood cells, bone cells, organ cells. But do all body cells divide? Of course not. Nervous cells and muscle cells, they don't do mitosis and are not renewed. Okay, so you have a cut in your hand. You put a bandage, and in few days, it's all healed. You don't think even about it. Or, do you look like now, like when you were 10 years old? Of course not. You are much bigger now. Why? It's mitosis. It's important for you to grow and also to repair your cells. However, sexual reproductive cells, like sperm, like eggs, they don't do mitosis, but they undergo meiosis that will be discussed later in chapter 9. So what's the role of mitosis? It has two important roles. The first one is the development and the growth. The number of cells increases by mitosis, enabling, for example, the zygote to develop into a human being. Also another role, which is the cell replacement and renewal. Cells are constantly lost and replaced by new ones in the body. For example, we renew our skin every week. We renew our red blood cells every 4 months. What is a cell cycle? It is the series of events that take place in a cell that cause it first to divide into two daughter cells, and these events include, first of all, the interphase, which is the duplication of DNA, and mitosis, or which is also called cell division. So the cell cycle is the combination of interphase and mitosis. What happens during interphase? The daughter cell, which could be also the zygote that is formed from the sperm and the ovule. So this daughter cell grows up, as you see here, by a process that we call interphase, and becomes a mother cell. And this mother cell is now ready to start the process of mitosis that, as you see, it's a process of division into two daughter cells. So, of course, we're going to start together by interphase to see how does a daughter cell or how does a zygote grow up into a mother cell. so it can later on divide by mitosis. Interphase. What is interphase? It's the period of growth. And during the growth phase, what happens? Look at the figure, please, here. This is a cell at interphase. The chromosomes represented in red and blue are long, very thin filamentous, and we call them chromatin material. So, if you look at these chromosomes very well, are you able to count them? Of course not. You can't count them because they form a mass that we call chromatin, made up of very long and very thin filaments of DNA. So this cell in interphase has the chromatin found inside this nucleus. The nucleus has a nuclear membrane and contains a structure that we call nucleolus. And of course, it contains the DNA that is represented by the chromatin or the long, thin chromosomes. In the cytoplasm, you see that there is an organelle that we call centrosome. The centrosome is made up by two pairs of centrioles, and the role of the centrosome is very important during mitosis. We're going to talk about it in the next slides. So, a main process happens during interphase. This process is the DNA duplication. Actually, at the beginning of interphase, every chromosome has only one chromatid. At the end of interphase, every chromosome now has two chromatids. In blue, this is a chromosome with one chromatid, and this is the second chromatid. The other chromosome in red, one chromatid, and this is the other chromatid. So, the cell with two N chromosomes, because we have a pair of chromosomes, so we say 2N, two chromatids per chromosomes that are long and thin is now ready for division by mitosis. What is mitosis? Definition of mitosis: it's a process of cell division which results in the production of two daughter cells from a single mother cell. The daughter cells are identical to one another and to the original mother cell. Mitosis is divided into four stages, which are prophase, metaphase, anaphase, and telophase. Let's start first of all by prophase, and we're going to consider the mitosis of an animal cell where 2N equal 4 chromosomes or two pairs of chromosomes. So this is the cell in prophase. So the cell that is here in interphase with long and thin chromosomes with a centrosome now starts the first phase of mitosis, which is prophase. Okay, what happens during prophase? First of all, chromatin, long and thin chromosomes, they condense into short and thick chromosomes, every chromosome made up of two chromatids. So this is a chromosome of two chromatids. So the long and thin chromatin becomes short and thick chromosomes. Now they are visible and we can count them. Okay? So what is the meaning of condensation? Condensation means thickening of chromosomes, long, thin into short and thick. Then the nuclear membrane and the nucleolus, they begin to disappear. Of course, they start disappearing progressively. The nuclear membrane is still visible here, but it's going to disappear progressively. Then the centrosomes duplicate and transform into two asters. So this centrosome transforms into two asters, and the asters, you can see there are spindles or spindle fibers between these two asters. So, second phase, mitosis of an animal cell, always with 2N chromosomes or two pairs, but now we are at the second phase or metaphase. What happens during metaphase? You can see clearly here that the 2N chromosomes or the 4 chromosomes, 1, 2, 3, 4, they are aligned at the equator of the cell, forming what we call the equatorial plate. Every chromosome fixes on one spindle fiber by its centromere. So you see, this is a spindle fiber. Every chromosome fixes on one spindle fiber by its centromere. So the 2N chromosomes or the 4 chromosomes are now aligned at the equator of the cell. And of course, the two asters are now at the opposite poles of the cell. So the cell has always 2N equal 4 chromosomes. We move now to the third phase of mitosis, Anaphase. So what happens in Anaphase? The two chromatids, they separate from each other due to the division of the centromere. So the centromere divides into two centromeres, and every centromere carries one chromatid, forming one chromosome of one chromatid, and moves to one pole of the cell. So we have now, instead of having 4 chromosomes here in the middle, we have now 4 chromosomes up and 4 chromosomes down, and every chromosome is now simple, made up of one chromatid. So in Anaphase, we can start talking about two cells, an upper cell and a lower cell. The upper cell has 2N equal 4, and the lower cell has 2N equal 4 chromosomes. Remember, every chromosome is now of one chromatid, so the genetic material has divided during Anaphase. So this Anaphase is characterized by what we call polar ascension. What is the meaning of polar ascension? It means migration of the chromatids or of the simple chromosomes toward opposite poles of the cell. So it's when the 2N chromosomes of one chromatid move to the upper pole and the other 2N chromosome of one chromatid move to the lower pole. So the last phase, which is telophase. What happens in telophase? You see here the cell that is elongating, preparing itself for division. First of all, the chromosomes that were thick, they decondense and become longer and thinner. And this is the opposite what happens in prophase, where the chromosomes condense and become short and thick. Then the nuclear membrane reappears. So you see here, at every pole, we have appearance of the nuclear membrane around the chromosomes. Spindle fibers that arise from the asters, they disappear progressively. The asters that change back into centrosome, and then the cell divides due to this constriction into two daughter cells. Every daughter cell contains two N chromosomes, and since the example here is 4 chromosomes, so these two daughter cells contain each 4 chromosomes, just like the mother cell. But of course, chromosomes are here in the chromatin form, long and thin. So the two identical daughter cells can now start a new cell cycle that begins by interphase. Again, these two daughter cells grow up into mother cells, and every mother cell is ready to start again a new mitosis. So, to conclude, mitosis has a genetic significance. It's a process during which the mother cell transmits to each daughter cell a copy of the genetic information. It ensures the transmission and the conservation of the genetic information and of the number of chromosomes from one cellular generation to another. So, when we say mitosis, we can say there is conservation of the genetic information and of the number of chromosomes. Thank you, dear students. Please review very well this activity, and I'll be waiting for your questions.